What is diphtheria? In fact, let us try to understand and how this happened. Currently, we have very less number of cases of diphtheria, but before their vaccinations against these types of disease, the person have symptoms of sore throat, low-grade fever, malaise and hoarseness of voice. The bacteria can release their toxins that travel through the hematogenous route and affect various organs. The diphtheria have two types of presentation in fact. They have their sore throat presentation and respiratory involvement. Hello friends, our today's topic is Corynebacterium, in which we will discuss about a very important species, that is Corynebacterium diphtheriae. They cause diphtheria. They are gram-positive bacteria, but we can use some special stain to visualize the presence of these bacteria. The stain is Albert stain. Here we can see the presence of these bacteria. Bacteria is gram-positive bacilli having changeable shape, pleomorphic rod shape, often seen in V form or Chinese letter pattern. Because of their unique binary fission pattern, we can grow these bacterial colony on blood agar, CTBA agar, cysteine tellurite blood agar medium is a selective medium where the colony of bacteria show black appearance because of tellurite reduction. The bacteria causes diphtheria and we have control over this disease because of universal immunization program. We provide vaccination to each and every child. We know about the DPT vaccine. Even after DPT vaccine, our universal immunization program have pentavalent vaccine. In pentavalent vaccine, we cover diphtheria, pertussis, tetanus and Haemophilus influenza type B and hepatitis B infections. This pentavaccine cover and provide immunity against diphtheria. We give this vaccination over 6th, 10th and 14th weeks of life of an infant. After that, we provide booster doses also. They provide immunity against the infection of diphtheria. What is diphtheria? In fact, let us try to understand and how this happened. Diphtheria is a disease and occurs because of toxins produced by the bacteria Clostridium Corynebacterium diphtheriae. These bacteria colonize over the tonsil area. They can grow nearby area and these bacteria have ability to release a very potent toxin. Currently, we have very less number of cases of diphtheria, but before their vaccinations against these types of disease, the person have symptoms of sore throat, low-grade fever, malaise and hoarseness of voice. Why? Because the bacteria colonize over the palate and tonsillar region. They develop a thick grayish membrane in the throat, that is pseudomembranous colonization. If we try to remove the grayish membrane, it is not possible to remove those membrane and bleeding occurs. It is very different from the candida infection where we can easily remove the membranes. These membranes are pseudomembranes. Our body develops these types of membranes to protect ourselves. As we know, in the Staphylococcus aureus that fibrin meshwork development and some neutrophil entrapment including bacterial colony forms a membranous layering, that is pseudomembranous presentation. Our body produces our immune cells in response to these types of infection. Therefore, patient have presentation of cervical lymphadenopathy. Nearby lymph nodes get swollen. The appearance is bull neck appearance. This is colonization of Corynebacterium diphtheriae. The localized immune response and colony of bacteria develops a condition that have sore throat-like presentation, low-grade fever and malaise, hoarseness of voice-like condition, and the patient may have difficulty in swallowing and eating. The bacteria can release their toxins that travel through the hematogenous route and affect various organs like our cardiac muscles, and the presentation can be of myocarditis. The inflammatory response and interaction of toxins produces this condition myocardium is involved? Try to understand. In Staphylococcus aureus, we studied about endocarditis, where only endocardium is involved. Here, complete myocardium is involved because of the toxins produced by the bacteria. Correlate the things. In Staphylococcus aureus, bacteria in actual produces their colony over the endocardium. Hope you remember that things. In Staphylococcus aureus, bacteria produces their colony over the endocardium. Here, myocarditis develop because of interaction of toxins with the myocardium, and our immune cells started inflammatory response at the site. The toxin can involve our neurons. Therefore, cranial nerves may be affected. The toxin can involve nephrons. In fact, tubular cells gets involved because of the toxins released by these bacteria, and the ultimate condition is renal failure. Recently, the presentation of diphtheria is very rare because of vaccination, but in a non-immunized child, if this type of presentation is going to happen, we have to collect specimen through the throat swab nearby the area of pseudomembranes. The diphtheria have two types of presentation, in fact. They have their sore throat presentation and respiratory involvement. They can also have cutaneous presentation. There are two types of diphtheria. One is respiratory diphtheria and other is cutaneous diphtheria. In respiratory diphtheria, the presentation can be of fever, malaise, sore throat, and cervical lymphadenopathy. Respiratory diphtheria involved through the formation of pseudomembranes. They can cause airway obstruction, myocarditis, and cranial neuropathies. Airway obstruction because of direct colonization of bacteria. Myocarditis and cranial neuropathies occurs because of toxins interaction to the myocardium and cranial nerves. The other type of diphtheria is cutaneous diphtheria, where a punched out lesion with necrotic surfaces, where we can find chronic ulcer with gray membrane formation. Punched out lesion is a very important term related to cutaneous diphtheria. It is a chronic presentation having ulcer with gray membrane. Try to recall our today's topic. We have discussed earlier that gram-positive bacilli have their presentation of spore formation and non-spore formation. In non-spore forming bacteria, we include branching pattern showing bacteria and non-branching pattern showing bacteria. In non-branching, we include Corynebacterium and Listeria. Corynebacterium diphtheria causes diphtheria. Two types of presentation, whereas gram-positive bacilli having non-spore forming tendency, non-branching pattern. Two examples, Corynebacterium, diphtheria and Listeria. We studied about the Listeria also, that they can involve adults and newborn. The infection acquired by a newborn can occur through the mother. The newborn can involve, and the ultimate presentation can be abortion, preterm birth, meningitis. This bacteria can cause bacteremia and meningitis in adults. In healthy individuals, they can cause gastroenteritis and watery diarrhea. It means we have to recall these two bacteria together. Corynebacterium diphtheria and Listeria monocytogenes. Corynebacterium diphtheria, how becomes that much virulent? They become virulent because of their toxins that involve myocardium, nerves, and renal tubules. What toxin do? Diphtheria toxins, in fact, inhibit the protein synthesis in our cells. Therefore, cellular death occurs. Diphtheria toxins have two subunits, subunit A and subunit B. Subunit B bind to the toxin receptor present on our cell. This is our cells where toxin is going to bind. The subunit B is going to bind over the toxin receptor, and receptor-mediated endocytosis occurs here. After that, the actual subunit of toxin, the subunit A, is going to release into the cytoplasm. This subunit modify the elongation factor. In fact, the subunit toxin, subunit A, modify the elongation factor necessary for the protein synthesis. As we know, over the mRNA template ribosome gets attached, and because of the interaction of these two, transfer RNA, transfer their amino acids, and ultimate result is binding of amino acids and formation of peptides bond, and a long chain of amino acid is going to develop. For the development of these polypeptide chain or peptide chains, we need some other factors. The factor is elongation factor 2. The toxins subunit A target this elongation factor 2 and they modify their shape. There is ADP ribosylation of elongation factor 2, and because of this modification, there is inhibition of protein synthesis or polypeptide chain formation, and the ultimate result is death of host cell. It means the toxin can kill our cells. Therefore, we can conclude that the virulence factor of the bacteria is diphtheria toxin. It is primary virulence factor that inhibits the protein synthesis by ADP ribosylation of elongation factor 2. This is a very important statement. The other important statement is that the genes associated with diphtheria toxin is encoded by a virus, not by the bacteria. The toxin is encoded by beta phage virus. The other virulence factor of the bacteria is adhesion factor. They can cause biofilm formation for their survival. Because of biofilms, they enhance persistence in host tissues. How the bacteria transmit from a person to another person? They can transmit through respiratory droplets. There can be a possibility of direct contact with infected individuals, like the cutaneous diphtheria can involve the inoculation to other person. The contaminated objects can be a method of transmission. Asymptomatic carrier can also transmit this disease. And it is a very interesting fact, by the vaccination, we develop immunity by providing training to our body. Try to understand. For the vaccination, we do not use the whole bacteria. We generally receive the toxins produced by the bacteria and we change them into a less harmful form that is known as toxoid. And this toxoid is a source of vaccine. Therefore, our body develops training against the toxoid or toxins. We develop antibodies against the toxins or toxoid, not against the bacteria. Therefore, even after vaccination, our skin or throat may have the colonization of these bacteria because our training is against the toxins or toxoid, not against the bacteria. Asymptomatic carrier can also spread the bacteria. What is the clinical presentation? We have already discussed about this. How we can culture these bacteria? We can culture on some selective media, that is CTBA agar, cysteine tellurite blood agar. In the microscope, we can see gram-positive rod, or to visualize the bacteria, we can use Albert stain, where the bacteria can show a Chinese pattern like or V-shape like their forms. We can also demonstrate the toxins produced by the bacteria. For that, we can use ELK immunodiffusion test, where we can detect the diphtheria toxins. Through the PCR assay, we can demonstrate or analyze the toxin producing genes. To kill the bacterial colony over the skin or over the tonsillar region, we can try antibiotics. Once bacteria develop their colony, they release their toxins, and the toxin is the main reason to cause infection or clinical presentation. Therefore, we have to give antitoxin therapy. The antitoxin therapy should be given as soon as diphtheria is suspected. In fact, currently the incidences of diphtheria is very rare because of diphtheria vaccine. The diphtheria vaccine generally provided through DPT vaccine. Currently, we have many other variations like Penta-Vac vaccine that not involve involve the DPT, they include the hepatitis B and Haemophilus type B also. Even some other vaccination combination may include poliomyelitis also. It means Infarix hexa vaccine provide immunity against DPT, hepatitis B, Haemophilus type B, and poliomyelitis. This is injectable form of vaccine.